Right, morning will begin. Last we spoke, we're doing this. We're trying to just learn this terminology for this concept map of the nervous system. And what we said for this example is that it was visceral motor. Call it example 1. Just to remind you of that, it's because it's a neuron carrying signals away from the CNS to the biceps. That makes it motor. And because it's innervating a muscle, I'm sorry, gosh. Let me wake up. It's somatic motor, not this one. Because it innervates skeletal muscle, not smooth muscle, it makes it somatic motor in its function. We're kind of going through all these different terms. So as a kind of a mental exercise to wake us up this morning is read the next one. The neuron carrying signals away from the CNS to the heart, increasing the heart rate. Take a minute to think which kind of motor fiber it is. I'll give you a minute, I'll check back in. Visceral motor, sympathetic. Why is it motor? Carrying signals from the CNS to the heart. Why is it visceral? Well, what's the heart? The heart is not skeletal muscle, so organs or contents or glands, that makes it visceral. And why is it sympathetic? Because I told you it increased the heart rate. That's one of the clues for the fight or flight division. So if it's autonomic, that means you have to decide if it's sympathetic or parasympathetic. Visceral means autonomic. You feel a hot candle burning your skin. I'll give you another minute, think about that. Somatic sensory. How do I know it's sensory? What was the clue? You feel something. Okay. You feel something, something is going from the periphery up to your brain, to your awareness. How do I know it's somatic? I said it was skin. Your skin is the somatic sensory part. Okay. You void your bladder during urination. What do you think that's going to be? Let's do this one together. This is how you reason it. Motor or sensory, that's the first category. When you go, is that a motor command or is that something you feel? It's motor. Okay, now I'm talking about the urinary bladder. So would that make it somatic or visceral? If it's the bladder, that would be visceral. So we know it's visceral motor. You may not know this. You'll learn it more in 431. Voiding your bladder is a sympathetic response. Holding it in before you go to the bathroom is parasympathetic. You'll learn that next semester. So visceral motor, sympathetic. This example. You feel the bran muffin you ate digest in your stomach. So again, you feel it, it's a motor or sensory. Yeah, that's what I'm going for there. Well, if it's sensory, what are you feeling? Your stomach churning from eating. Those sensations are visceral sensory. Switching topics, that's the terminology. Going back from gray matter to white matter, what does nervous tissue look like under the microscope? We give the example of neurons. Gray matter is gray in nervous tissue because of neuron cell bodies. Basically. And white matter is white because of the myelination in the axons. Let's talk about the histology. You can see gray matter and white matter with the naked eye when you look at nervous tissue. But in terms of the histology of the cell types, the neurons and the glial cells, I kind of talked about neurons already. We talked about all the different kinds and different parts, the motor neurons, the sensory neurons, multipolars, unipolar, bipolar, those things. The essential thing is that they have areas of input and output in our excitable cells. But these cells need a lot of support. So we'll go through some of the glial cells. They're supportive. They actually outnumber the neurons something like 10-1. So they're more numerous. So they're supportive, more numerous. And they, well, basically they're non-excitable. They don't conduct electrical nerve impulses like the neurons do. Non-excitable. They have important functions, and I listed a few on the slides that you need to know, including well, here's a list of them, but we'll go through them one by one, starting with these astrocytes. So glial cell types. First one, those are astrocytes. They help recycle neurotransmitters, but the first thing I put is the more important thing. They have little processes, they call them perivascular feet, because they wrap around capillaries. Capillaries are the blood vessels that allow exchange of metabolites and gases in your tissues, and you want to allow that. That's their normal function. But in nervous tissue, you want to restrict that exchange. And they call it the blood-brain barrier. Whole blood is really toxic to nervous tissue. So these function to form the blood-brain barrier, as we call it. Perivascular feet wrap around the blood capillaries forming the blood-brain barrier. Well, this is a barrier between the whole blood that's in the blood vessels and the CNS interstitium. So that's what they call it. They use the word barrier. It's a barrier between whole blood and the central nervous system, the CNS interstitium. So you're just limiting that capillary exchange. That's a normal function of capillaries. And so, yeah, that protects nervous tissue from whole blood. This is a picture of a microglial cell. They're basically like the macrophages of nervous tissue. They just keep things sterile by phagocytizing microorganisms and pathogens. Alright, so that is microglial cells. How are they phagocytized? Things sterile. Things sterile. Next up, you have ependymal cells. These, they look like epithelial cells. What they do is they form a membrane. This is another barrier. This is a barrier between blood and CSF. So here is kind of where you get the formation of cerebral spinal fluid. So they form a membrane, and what they do is, what is not really showing you there is there's tight junctions between those cells. So all right, there's tight junctions between ependymal cells. And again, you're limiting that, you're filtering the blood in a way that you filter out all the stuff that's toxic to the brain. And so at the top of that figure, that blue stuff, that's supposed to be the fluid-filled cavity that is CSF, the cerebral spinal fluid. So for blood here, filter it through there. Only very small molecules can get through, and then you end up with blood that's been filtered into CSF. So I'm just writing there are tight junctions between the ependymal cells. They basically filter blood and then form CSF, which stands for cerebral spinal fluid. Cerebral spinal fluid. actually two words, cerebral spinal fluid. Put a little more space between there. Or CSF. Well, this is important because it's CSF that circulates around the central nervous system and nourishes nervous tissue. It's not blood. And you have basically two locations where this occurs, this blood CSF barrier. That's what it's called, blood CSF barrier. So not to confuse it with the blood-brain barrier. Which is a barrier between blood and the interstitium. This is a barrier that actually produces the cerebral spinal fluid. I'll put another note. The central nervous system is bathed by the CSF. CSF completely surrounds and circulates the central nervous system. We actually talk a lot about that when I talk about the brain at the end of the course. I'll just move on from now. Really, you're just supposed to know the epidermal cells and how they form CSF for now. Now, the oligodendrocytes, they myelinate the axons of the central nervous system. That's all I want you to know. So they show the myelination of these telephone cables, the axons, as we call them. They're the cell bodies. So the myelination is right around here. That's just the cell body. But the processes for the myelination. So, that's all right. Oligodendrocytes. There's a couple of cells here. One are the satellite cells. They hover like moons around the cell body. That's why they're called satellites. But they recycle neurotransmitter. And then you have the Schwann cells that myelinate the axons of the PNS. So the oligodendrocytes myelinate axons of the CNS, and it's the Schwann cells that myelinate axons of the PNS. So let me write that down. The next slide just shows the folding of the Schwann cells cell membrane around the axon, thus myelinating it. So myelination is a fatty wrapping of the plasma membrane around a piece of axon. So Schwann cells myelinate axons of the peripheral nervous system. Here you can see a myelin sheath around an axon. It has many layers. If you look at the top of the slide, it's labeled. Here's the diameter of the axon, it's a cross section. So all these layers are the Schwann cell wrapped around it. All right, so we move into the next batch of slides. It's called membrane potential. And this is actually a review of concepts that we learned before in the first chapter when I talked about membrane transport. So I'll talk about, we're going to get back to the neuron. on physiology. So what I tried to teach you in the first lecture is that these cells are excitable. They have all these areas of communication, areas of input, areas of output. So we're going to kind of get into that, this excitable cell idea. So if you're excitable, you can be in two states. You can be not excited, call it rest. Or I guess you can be stimulated. Well, if they call it resting membrane potential, obviously it's the first one. And here they show you the idea of resting membrane potential, and they show you like an experimental setup. Much like we kind of talked about when you did the muscle physiology lab. And here's our cell. There's a cell body. And let's see if you remember. Okay, these little processes here are the dendrites. Did we say that's area of input or output? This is area of input. What about this long one here? That was the axon. Was that the area of input or output? And this was output. And that's kind of where they're measuring it. So they like to study the area of output. Okay, well, we'll talk about how you stimulate the cell, but they kind of like to look at the area of output part of the cell and the cable part. And we talked about this before, where you kind of have this charge separation or membrane potential. The concept was membrane potential, and it being polarized. Or I don't know, you look at the top part there. And you have a charge separation. Negative, 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 right? And then positive, 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 positive. That charge separation, and it's like they're kind of attracted to each other across the membrane. That's the membrane potential. So if you were to measure that using your voltmeter, you have to put the electrode in two locations. Here's one location, and then the other location on the inside. So whatever the difference in charge is in the being measured by your two electrodes, you know, they say that it's negative 70 millivolts. So here's what we learned. That voltage is the difference in charge separation. Across the cell membrane. When you take freshman physics, you'll learn Ohm's law, which describes the electrical current voltage relationship. So V is voltage. It's what you measure. Okay, but it's equivalent to current times resistance. So in physics, I is current. Current is the flow of ions from one place to another. Well, in our case, it would be across the cell membrane. That's why we would be interested. In excitable cells, you kind of see this. Cells are permeable to different ions, and ions flow across the cell membrane. We kind of talked about that in the first chapter. Now R, what is resisting the ions from flowing? I mean, what is this double line thing I keep drawing? what is this supposed to represent? The cell membrane. So the resistance to ions flowing is the cell membrane. Why? Because it's got that hydrophobic zone. Ions don't like to flow through it. We learned that again in the first chapter. So R is resistance, but in our situation, cell membrane. So R, resistance, cell membrane. So if you could decrease the resistance by putting an ion channel in there, current would flow and you'd see a change in voltage. So that's how this equation makes sense. We're not going to really apply the equation. Take physics and you'll do it. Okay? But just the idea of why do we measure voltage? And that's what we could do and we could see it. And so we call this polarized because this is not equilibrium, right? This is negative 70. If you want that to read 0, you have to like make it, I did this before. Okay, that would be 0. That would not be polarized. And most non-excitable cells, this is kind of what you see. So this is actually special. And we talked about it before, how the cell is able to maintain this polarized state. Because what you should see is that you have potential to kind of like be excited by allowing currents to flow or to depolarize. This you can't stimulate. Nothing's going to happen. It's already in equilibrium. So this is special, and this is our rest. So a lot of students, a mistake students make is they equivalent, they say for rest, they call this equilibrium because rest and equilibrium sound like the same concept to you. This is not equilibrium. This is, okay? So don't make that mistake. All right, so a neuron at rest has an excess of positive charges on the outside, as I've drawn here, and an excess of negative charges on the inside, as I've drawn there. And we measure that to be negative 70. This resting potential is established by many potassium lead channels. Establishing rest, this negative 70, is as easy as 1, 2, 3, A, B, C. And I kind of use this as a memory jogger for myself to remind you what I taught before. We'll go through that. So I'm going back to the first test. So call this, established a review of establishing resting membrane potential. So that's potassium. Potassium lead channel, sodium lead channels. I'll draw one sodium. Sodium lead channel. Let me draw a couple more potassium lead channels. Remember studying the sodium, potassium, ATPase pump. camping, but that was on the first test. What we had talked about at the time was all of these are embedded in the cell membrane. And I drew more of the potassium lead channels because these cells are more, they use the word permeable, they're more permeable to potassium. So I drew more of them than they are to the sodium. So I drew, fewer sodium lead channels. And what we had said earlier is that the cells, they work hard to make it so that there's, let's say this is inside, this is outside. That inside a cell, the gradient is such that there's more potassium on the inside, concentration-wise, than there is on the outside. So I'll draw my chemistry concentration brackets. There's more potassium on the inside. So the gradient is such that potassium tends to leak out. Okay. And we said the opposite was true here, right? There's more sodium on the outside, and sodium tends to leak in. So there's more sodium on the outside. There's far less sodium on the inside. So there's less leak channels, but it tends to leak in. That's kind of what I said there. So I think I said why it's easy as 1, 2, 3, A, B, C. I want you to remember 1, these leak channels, 2, these leak channels, and 3, the pump that counteracts the leak. Because what we had said before is you don't want the leak channels to dissipate the ionic gradients. Okay, that's a concept I tried to teach you before. We don't want the leak channels to destroy ionic gradients. Ionic gradients. So my ABC is how the pump works, okay? And, well, let me advance the slide here. At a ratio of 3 to 2, sodium's leaking in, so we pump it back out. Let me draw a couple other ones here. Sodium's leaking in, and it would destroy the gradient of sodium coming in, so we want to pump it back out. So I don't know, imagine these 3 being pumped back out. I think your book gives an analogy of guys in a fishing boat, but there's a hole in the boat. Okay, there's a couple of guys, maybe one guy is holding a fishing pole. And another guy, I can't draw. In the analogy, they say, because there's a hole in the boat, water is coming in the boat. And this guy's job is to bail out. So in this analogy here, sodium is supposed to be like the water coming in the boat, and you want to pump it back out. Okay. So, bail out water or sodium. I hope this analogy helps you. It's kind of helped me remember over the years. Okay, this guy, it's like you have your catch of fish, Here's your fisherman's net, and it's full of fish. But because there's a hole in the boat, the fish escaped, and they're like swimming away. So this guy's job is to catch the fish that are escaping from the boat. And so you kind of put a little hook in their mouth there and try to catch it. So that's supposed to represent this. It's leaking out, but you want to like pump it back, get it back in the boat. So you take 2 of these, and you pump it back in. Because remember, it's a 3-to-2 ratio, this pumping exchange. So the end result of all this is you maintain these gradients. Let me write this. By the action of the pumping of the sodium, potassium, ATPase pump, you counteract the forces of leak, and you maintain your resting membrane potential of -70. Counteract forces of leak, maintaining resting membrane potential -70 millivolts. Different books might use a different number, -65, -75, but something like that. The inside of the cell is negative with respect to the outside. Okay. So everything I did here should have been a review, but are there any questions on it? This is kind of how we talked about before. You're establishing why the inside of the cell is resting at a negative state.
What is the difference between the electrochemical gradients and ionic gradients?
I didn't hear the first part. What's the difference between what and what?
Ionic gradients and electrochemical gradients.
Okay. We can talk about that. Good question. That's actually what I want to get into now. They're basically the same thing, because an ion has a charge, which makes it an electrochemical gradient. But let me kind of...
Because in this case we can say ionic gradients. That's not
Yes, because ions carry a charge, you can also think of it as an electrochemical gradient. And this actually starts to address that point. So what I want to do here is, because we consider sodium and potassium at the same time, but let's consider it one at a time. We can talk about electrochemical gradients of sodium and potassium separately, then together. So let's first consider the electrochemical gradient of just potassium. And sodium's not even here. And they happen to use the same orange bulbs. I use orange for potassium too. Okay, so electrochemical gradients of potassium. Why do we even talk about this? Because the neuron is permeable to potassium. It has all these potassium channels, and so we have to talk about it. Okay, why would a cell be permeable to ion? It must use it for its physiology. So let's just talk about why potassium is so important. Okay, we need to eat potassium, and our cells need it. So what they're showing you here is this gradient, this -70. And All right, well. Let me explain the arrows. This is the chemical gradient, this black arrow, and they point it out because just considering the concentration gradient, not the fact that potassium has a charge, but just the concentration gradient, it would want to leak out because there's more on the inside and you want to diffuse from high to low. However, the electrical gradient points in. So think of it this way. For every positive potassium that leaves the cell, you're left with one excess negative charge on the inside of the cell. There is one excess negative charge on the inside. So let me pause there. A positive potassium left the cell. So by default, by one leaving, pretend there's negative charges on the inside. Usually proteins will carry a negative charge. Okay, you're with me so far. One leaves, a positive leaves, no, by default there's excess negative. That excess negative charge now wants to pull the positive potassium back in. This excess negative charge, quote unquote, wants to. It doesn't desire. I'm just trying to get the concept across. It wants to pull the positive potassium back in. Back into the cell. So that's why in this situation, the chemical gradient is out because there's more on the inside than the outside. But so it's leaving. But every time it leaves, those excess negative charges, it wants to pull it back in. So that's why the electrical gradient, the arrow is pointing in the opposite direction. Now, one thing to notice about physics, they make the arrows different sizes. So which force is stronger, this chemical gradient or this electrical gradient? Which is stronger in this situation? The chemical gradient is stronger. So what you can assume is, well, more potassium will leave than get pulled back in. That's what you're supposed to assume. That's why they put this third arrow here. Well, what's the net electrochemical gradient? Well, it's out. So the question becomes that physiologists always like to ask, at what point do you reach, the equilibrium potential for potassium? Okay, so if you let potassium flow until those two forces are exactly equal, until chemical and electrical gradients are equal, you get the bottom picture there. They're equal and opposite. They call that the equilibrium potential for potassium. You've reached the equilibrium potential for potassium. So if you were to measure it, it's like negative 90 millivolts. So the equilibrium potential, it considers both the chemical and electrical gradients. The point at they're equal, negative 90. So the cell is very negative. So it's like, it's so negative, negative 90 to be precise. If one tries to leave, it gets pulled right, it's equilibrium. It's kind of balanced right there. All right, so we have to have the same conversation for potassium as well as for sodium. So for sodium, it's the same conversation. Here's the rest condition. The chemical gradient is in. So is the electrical gradient because it's already negative on the inside. So there's a net in there. So it's like, for sodium, it's like for every sodium that goes in, there's an excess negative charge on the outside. Okay, it's just the reverse way of thinking. So it's like, if you just keep allowing sodium to flow in, eventually, well, here, let's just cut to the chase. If sodium keeps going in, so that the inside is so positive, it won't want to go in anymore. So that's where these two forces are equal. Okay, so it keeps going in, it's so positive on the inside of the cell, which means there's so much negativity on the outside. Sodium can't diffuse in anymore, so these forces are equal. The equilibrium potential for sodium is plus 66. Let me write that down. Equilibrium potential for sodium plus 66. So at the very least, if this were a nutrition class, you know you need sodium, potassium in your diet. Your cells need it. In terms of how the cell works, boom, plus 66, negative 90. Those are the equilibrium potentials for sodium, potassium, respectively. What was our resting membrane potential? What was the number? Negative 70. Okay, which is it closer to? This number, which is it closer to? It's closer to that. So the cell lives at rest much closer to potassium's resting membrane potential. So what that means conceptually for you to know is that cells are more permeable to potassium because its rest is closer to its equilibrium. Neurons are more permeable to potassium. So that's the condition of rest, just negative 70. You can be stimulated, okay, but you rest at this very negative potential, okay, almost at potassium's equilibrium potential. So when you stimulate it, you can depolarize that negative state. So here's what I charted out on the board earlier to show you that at negative 70, those leak channels are going, but for every 2 that leak out, they're pumped back in. For every 3 that leak in, they're pumped back out. The pump is counteracting these passive forces of leak. Okay. Here they show that proteins also contribute to the negative charge. That negative 70, that's worth noting. You see that everywhere. Proteins. The pump, sure, but yeah, proteins contribute to that negative 70. Proteins carry negative charges. So I'll just make the connection. That idea helps contribute to the negative resting membrane potential of negative 70. Here's how the Maria book kind of goes about it, by showing you one at a time. First, that the leak channels, they're more leaky to potassium. So they show a couple of potassium leak channels, and we know that the resting membrane potential, negative 90. Then they add in a leak channel to sodium, okay? That brings it up to negative 70 because positive sodium is leaking in, bringing up your membrane potential. But then they add the pump because that will counteract these forces of leak so the ionic gradients don't dissipate, don't get destroyed. So that kind of sums it up, I think. Let's take a break, and we'll get into excitement there. Come back to 8:45.